Welcome to Bioinformatics Basics. In this episode, we're taking a very simple look at DNA sequencing. And don't worry if you're completely new to bioinformatics. No prior knowledge is required. Let's start with DNA. DNA stores biological information, and it's made up of 4 different bases: adenine, thymine, cytosine, and guanine. We usually represent them simply as A, T, C, and G. So, what exactly is DNA sequencing? Simply put, sequencing is the process of determining the order of those bases in a DNA molecule. Instead of seeing a sequence we don't know, we're working to determine exactly what that sequence is. But why do we want to know the DNA sequence in the first place? Because the sequence contains information about biology. Sequencing can help researchers study genes, microorganisms, genetic variation, disease, and even how organisms have evolved. And this is where bioinformatics comes in. Modern sequencing can generate huge amounts of data, including millions of DNA reads. Computers and bioinformatics tools help us process and analyze that data, so we can turn raw sequences into useful biological insights. So here's the key takeaway. DNA sequencing tells us the order of bases in DNA, and bioinformatics helps us make sense of the data that sequencing produces. So now we know where sequencing data comes from. But what does that data actually look like? Next, we'll take a look at something you'll encounter very often in bioinformatics: the FASTQ file. Follow the digital genome as we continue exploring genomics, bioinformatics, and the science behind biological data. The digital genome: learn, analyze, understand.